Oh, am I we actually talking about it? Yeah. Okay, quite you.
Okay, let's get back for a very exciting addition to this year's event. And it's another collaboration. Just like the morning, we expanded our topic a little bit to the Mesa to include Salk and Scripps Research Institute. So this one was really sparked by a conversation I had with Marianne Beister a few months ago at an event. I was telling her about this event, and she, as always, is very passionate about the environment. And she was like, okay, we need to figure out a way to connect the environment into this. And SIO is one of the crown jewels of UCSD. And we started talking about this and the whole notion of environmental and human health. So that's the angle we are bringing this under. And once again, I want to thank Marianne for that idea. And Marianne Beister, of course, needs no introduction here on campus. She's working with multiple units on campus. She was on the Dean's Advisory Committee. I think you continue to be in the Dean's Advisory Committee in various schools, School of Management and the...
Yeah, I rolled off at the Rady School, but I'm on the Director's Council at Scripps.
At Scripps, and several of campus leaderships tap her as an advisor. So we're really grateful to her for orchestrating this panel and introducing our star inventors, and we can have a discussion here. Okay?
Cool. These guys? All right, hello. Hey, can you hear me? This works? All right, good. So I want to thank Vish and Charlie for bringing this all together. Vish, our relationship goes back about 20 years since the founding of the Rady School, so it's been fun getting you know through lab to market initiatives. And Charlie, we've met longer than that. Back east at our with a gathering. I think it was a party actually at the medical school when I was in grad school close by. So I'm really excited for what you guys are doing. I know that your collaborations and connections have only deepened and looking at how we can improve innovation in the healthcare industry. And now we're here to describe a little bit more about how that connects to oceans. So let me give you a little bit of background of Scripps Institution of Oceanography, which today I'll be saying Scripps and SIO. So when I say Scripps, I mean Scripps Institution of Oceanography. So we're a department at UC San Diego. We're actually the oldest. We existed before there was a UC San Diego. And we're a global leader in oceans, earth, atmospheric, and climate science research. That's both applied research and basic research. Specifically related to human health, there's a lot we could cover. We're focusing today's discussion a little bit more on the microbiology and macrobiology discoveries that can help with addressing human health. But I also wanted to mention that our researchers do extensive research in understanding the threats and the human health impacts of environmental pollutants and climate change. So we're the go-to institution in the country and the world for that. So today, this is a SIO contingency. We have two professors and lab directors from SIO. We have Jack Gilbert and Paul Jensen. They both run labs in the Scripps Marine Biology and Biomedicine Center. And amongst other things, which I will not be going through, their various accomplishments and roles, because they're going to give that to you in their presentations and then also in our discussions. But I do want to give you a little context for why they were selected for this panel. And both of them are very deliberate in advancing science through not only their own extensive research, but through their labs and through their commitments for science translation to human health. Both have a very strong and deep interest in research on the ocean microbiology as a source of new discoveries, and with that will come along interesting new risks and surprises, which we will cover. And their training, their relationships, and their work is highly disciplinary. And when I talked to Charlie and Vish, I thought that was a very interesting aspect to bring to you all. What were the advantages and some of the challenges along the way? So we'll have introductory comments both from Jack and Paul, and then we'll go into a moderated Q&A. So maybe hold off on your questions for each of them, but then I promise I'll weave in your questions in the moderated Q&A. All right, so we'll start with Paul.
You go first.
Yeah.
All right, hi everyone. And so as Marianne mentioned, I'm at the Center for Marine Biotechnology and Biomedicine. And
It's also
Yeah, your voice is very soft.
okay, never been accused of having a soft voice before. That's okay. So who knew, right? So we're an oceanographic institution, but we're doing biomedical research. We're very engaged in drug discovery. And I want to tell you a little bit about some of those things in my quick 5-minute introduction here. And so, so with this audience, I think everyone recognizes the historical importance of natural products in terms of drug discovery. Okay, microbes have played a big role, most of our antibiotics. In fact, most of our small molecule therapeutics are in some way either derived from or inspired by natural products, okay? So these molecules remain hugely important, and historically, they've been a great resource. However, we've kind of hit a little bit of a wall in terms of how much we're discovering relative to the needs that are out there today. And so what we've learned through using genomic approaches is that even organisms that we've studied for 20 years in the laboratory, when we sequence their genomes, they have the potential to make many molecules that we have not yet found, okay? So even the organisms that we can culture, they can do a lot more than what we're finding. So there's room for improvement there. When we do biodiversity studies, we realize that most of the microbes on the planet we haven't yet cultured, and so we're missing a whole lot of opportunity there. And when we do metagenomics, we can sequence the genomes of those organisms that we've never cultured, and we can see that they have the potential to make all kinds of molecules that we've never found. So there's a wealth of chemical diversity out there that we have not yet captured and explored for its potential in drug discovery. So we've taken a couple of approaches to this. We need new approaches, that's very clear. We're very interested in going to the ocean and looking for new groups of microbes that might produce interesting natural products, and that's been an interesting activity for us. We've come across numerous interesting new groups. One, the genus Selenospora. All of these molecules come from that genus. It's a very rich source of new natural product chemistry. One of these molecules, top boxed in red, Selenosporamide A, is a proteasome inhibitor. It's currently in phase 3 clinical trials for glioblastoma. It crosses the blood-brain barrier, something that's not very easy to do for a small molecule. It's also being tested for multiple myeloma. So we're very excited about that. We find a new group of organisms, lots of new chemistry. So this is a valid approach, but even with this approach, we're still missing most of the potential, okay? And this is just one group of organisms that we can culture. There's so many more that we're missing. So this is the way it's always been done, right? You make a collection of microbes, you culture them, you grow them, you screen them, you look for drug candidates using that approach. So what we've been thinking about is, why don't we flip that approach? Why don't we go straight for the molecules and then try to figure out who's making them, okay? And the way we're doing that is we're putting adsorbent resins out into the marine environment, and we're capturing molecules directly from nature. We call this small molecule in situ resin capture. And what we can do then is we can take the molecules we captured, we can put them through various bioassays, in this case an antibiotic assay. We can follow the activity and isolate molecules that way. And so this is actually proven out to be a lot more successful than I thought it would be when we started. big question was, can we get enough material to actually solve the structures of these molecules by NMR unequivocally? And that was our big question mark. The answer is we can, and what we're finding is really extraordinary. We're finding incredible new carbon skeletons never seen before in nature. Here's one of the molecules that we found. It has some very unusual features, structural features. It's got a bromine and a chlorine, hallmarks of marine natural products, halogenation. It's got this feature here, circled in red, which has never before seen in the natural products or synthetic literature. It's a vinylous formate ester. And so we're seeing things that are totally unprecedented, that nature knows how to make. We don't. We've never before. And these provide great opportunities for testing. Here's another molecule, a completely new carbon skeleton. This one, we found some really intriguing biological activities. I'll just tell you about one. It affects the amplitude and frequency of cardiomyocyte beating. And so treated with a compound, you can see the amplitude increases, the frequency decreases like some heart medications. So enormous possibility for discovery. We think we have a way to now capture this wealth of chemical diversity that we believe is out there. And so, yeah, so we're going for it, very excited about this. This is agnostic to the producing organism, right? We don't have to target an organism, culture it. This can capture molecules from anything. We don't have to build large culture collections. When we do this, when we deploy these resins in different habitats, here you can see in this plot, each dot represents the chemical profile of one of the extracts. And you can see when they're from one location, they tend to cluster together, but the locations are very different. So there's very different chemical diversity in different marine habitats. There's an enormous amount of chemical diversity that we can access using this approach. So super excited about this. There are challenges, of course. Resupply is one. Happy to talk about that if that wants to come up in the discussion section. But I think I'll end with that.
All right, hard to follow. My name is Jack Gilbert. I'm, as heard, I'm actually the Associate Vice Chancellor for Marine Science down at SIO, Deputy Director for Research. But I also have a joint appointment in pediatrics. And I came from University of Chicago in the Department of Surgery for 10 years. So I really don't know who I am or what I'm doing most of the time. But I do study microbes. And I'm very engaged in trying to look at cross associations between environmental research and human health research, which can power both areas. I'm also the president of one of the oldest microbiology societies in the world. I just want to pitch this here. We are very, very engaged in applied microbiology, and we are really pushing for applied microbiology processes in universities across the country. One of the reasons I moved from Chicago to here was because UCSD has a very strong applied microbiology or applied scientific development program. So we were really keen to take advantage of that, look into opportunities for innovation in building small companies and providing opportunities. And AMI is a leading organization in helping to promote those activities at universities across the world. So that's just a pitch. So we heard about marine science. We do marine research, and mostly it's focused around trying to identify the etiological environmental origin of infectious disease, particularly in a changing climate. So focused around trying to see if, for example, oyster farms might be more susceptible to increased human pathogenicity in the face of rising sea temperature, microplastics, pollution, etc. But the other side of me, the pediatric side of me, is focused on human health. So I thought I'd pitch that as well. I'm very interested in that intersection between the immune system and the microbiome, the microbial community that encase our body. You each have about 2 pounds of microbes living inside you, about 40 trillion organisms, and they are managed by the immune system, which acts like a gardener, right? It's there to allow the organisms to thrive and survive, which produce products, like we heard from Paul, that help our health, right? So our body farms its microbiome to facilitate healthy development. And it will get rid of the ones that aren't effective or aren't helpful. But any disruption in either two of these sides of a coin will cause big impacts on the other. So if you disrupt the microbiome, you disrupt the immune system and vice versa. So we've been trying to understand how to manipulate the immune system, which we see as a central element to improving human health, because it intersects with hormones, intersects with neurodevelopment, intersects with cardiometabolic disease. We want to see that by manipulating the microbial community. One of the areas that we've been focused on is influencing neurodevelopment. So we have a big neuroscience program working with the biological sciences department here, school here. We're a department and a school. You have a school and many departments. It's very different. We do nuclear magnetic functional magnetic resonance imaging of people's brains, identify brain regions which associate with disease, for example, major depressive disorder, and then look for the bacteria present in people that co-associate and correlate with that. We found one organism, for example, or two species of Bacteroides that were highly associated with an absence of brain signatures associated with depression. So when you have this bug, you don't have any brain signatures of depression, you don't have any phenotype of depression. When you don't have this bug, it's there, and nearly 100% accuracy in most populations. And so using that, we worked with a very talented graduate student, Marisol Dardar, one of my postdocs, Promi Das, and partnered to form a company called Holobiome that's currently in Boston, and set up a mechanism to produce these bugs and form these bugs into a formulation which can be delivered preclinically and clinically. And so in a preclinical model of depression, for example, social defeat, this species is as good at alleviating the symptoms of depression as ketamine, right? So it's a potent antidepressant, but it has no side effects. It's an organism present there and needs to be beneficial for human health. This is currently going into phase 2 clinical trial later this year. We also are interested in understanding how we can monitor the microbiome, both in the environment and in human beings. And so I run a large-scale NIH program, a $200 million initiative called the Nutrition for Precision Health, which is focused on trying to understand how we can predict ideal nutrition for an individual based upon the microbiome, and ideally how we can see why people respond to nutrition so differently. So to understand the microbiome, you have to put it in the context of other features that we look at in human beings. For example, height, weight, and age, which are low-dimensional and very static. They don't really change. Well, they do. Your age changes, but not in a significant way on a daily cycle. Or insulin and blood pressure, which are low-dimensional, but they're highly dynamic. They change hourly. Or genetics, which is highly static, but incredibly highly dimensional, 23,000 human genes. The microbiome is like the worst case scenario for finding any associated features. It's incredibly difficult to find effective biomarkers of disease because it's highly dimensional and highly dynamic. So to discover these things, we need very big cohort sizes in our human population studies, and we need frequent sampling, which is something which hasn't been possible to date. So again, we formed another company called BioSense, which has produced a molecular biology laboratory in a box. And this device can basically sit in any environment and take a sample, and we'll extract the DNA and RNA and preserve it for up to 6 months. So you can imagine this, we are tackling an at-home poop collection market, where we want to collect all the fecal bowel movements from our participants in studies. But we're also deploying this, working with NASA to deploy this in lunar missions and in spacecraft missions, because it facilitates collection of material that doesn't have to be shipped back to Earth, or into field deployable trials in remote locations, so you can collect samples on a regular basis without to ship and store samples. So that's a little bit of a track into how we intersect with human health on that space.
All right, is this one working? Yeah. All right, I want us to go general, just make sure we're covering some basics, and then let you guys ask some questions and I'll ask some other questions. But I wanted to go back to the diversity of microbiology that we have in the oceans, and what you think is the most promising areas for that. It could be applications or, again, new analysis. But the most promising for advanced human health. Again, macrobiology could be, fish, algae. Let's just kind of broaden it out, because I know the breadth of your guys' understanding. So where do you see the biggest opportunities, And then also, what kind of relationships would be really helpful to take advantage of those opportunities, especially since you have researchers and clinicians in the room.
Okay, I'll start. You know, maybe just a little history, like down at Scripps, we've had a long history of some very clever organic chemists going out into nature and finding all these interesting, unusual molecules. What we haven't been very good at is translating those into medicines. And we're really good at discovering things. What we're not really great at is making the connections with people who know how to develop them. It's hard to get the pharmaceutical industry interested in a molecule unless you have, you know, a super advanced data set with all these activities and mechanisms of actions and all these kinds of things. And so, you know, the potential for finding new medicines in the ocean remains very great. There are currently 12 molecules that are approved for cancer, pain relief, some other indications. There are undoubtedly hundreds, maybe thousands more that are there. And so I think we're really good at the discovery part. Where we could use help as a department, I think, is to figure out better ways to try and take those discoveries to the next level and get the interest that's needed to advance them. So the one molecule that I told you that's in phase 3 clinical trials, we actually started a small company called Nereus Pharmaceuticals. They raised $200 million over the course of 10 years, spent all that money, got the molecule into phase 2 trials, then they went out of business. Bristol Myers ended up buying the whole package and they put it into phase 3 now. And so, you know, the barriers for doing these kinds of things are very high and difficult for us to achieve. So it would be great if we could connect the dots a little better. Like I was saying to Marianne earlier, you know, a lot of people don't even know we're doing drug discovery down in Scripps. And when we talk about it, we always show this picture of us and the rest of the campus and say, hey, we want to find things and do the pharmacology here and take them all the way through clinical trials here, which could happen, right? And it's never happened. That stuff's gone off campus to do. And we talk about that and we feel it's possible, but it's not always clear that the rest of campus knows that we have that interest and are thinking about, you know, going in the other direction.
I'll echo that and augment it just by stipulating that from my perspective, as we heard from Paul, the oceans, the sediments of the world, and even the organisms which live in those environments are incredibly diverse. There's an enormous amount of biotech potential, but it is just potential, right? Identifying which organisms are relevant is a monumental task. We need significantly better screening systems to be able to do that. One of our very talented professors is also in the same center as Paul and I, Amro Hamdoun, has developed, for example, a new echinoderm, isn't it echinoderm? Yeah. Echinoderm model system that uses echinoderms as larval genetically modified prediction systems to look at drug screening potential for understanding the impact of drugs on developmental processes. That's the kind of technology we do need. We need high throughput screens to get through the vast majority of the information that we can actually access. Otherwise, we're just collecting data. You know, I launched a program called the Earth Microbiome Project, and we worked with Craig Venter for many years on oceans microbiome research, and we've got enormous data resources. And like Paul was saying, you know, huge metagenomic data that tells us all the diversities there. We can find, you know, millions of biosynthetic pathways and genes and functional traits, but actually getting them into a system where we can determine what they actually do and how valuable they are is a monstrously difficult filtering process where we're leaving an enormous amount of potential on the table because we don't have the screens in place that facilitate us identifying whether they're valuable. So I'd like to see better innovation in those practices. Both Paul and I have obviously presence in SIO and on main campus and in other entities. And I think that's a really, really strong way of enabling people to talk to each other and help to develop engineering solutions, help to look at biosynthetic pathway identification solutions, big computing to allow us to get through the data. Those are really valuable ways of doing things, but we need those partnerships in place. And to a certain extent, we need the relationships to be mandated, right? So people don't have to work hard to build them, but they're part of the fabric of the ecosystem. And UCSD does a good job of doing that, especially in the last 10 years. And our Chancellor's done a great job at helping us to partner with different departments and breaking down the barriers and silos that would prevent us from making those relationships. But it's still not in the DNA necessarily of the academic sector. So allowing those partnerships and allowing public-private partnerships to thrive from those, I think, is the area where we need to focus on most.
So is there any question or maybe response from the audience that would connect on how to address some of the gaps that we have as crystal oceanography? Any, before I move on to a different topic? Anyone? Yes.
Thanks for great talks. I'm Arjen Fusting from Novo Nordisk. So yeah, it's exciting mounting in front of you of things that potentially you can do. Paul, from all this novel chemistry that you're getting, you showed the IPS, the cardiomyocytes. Like how are you defining? How are you getting those in vitro systems? How you decide what type of screening assay you're gonna have you decide to focus on a particular area, for example, and then get the strength and try to narrow down in that before moving into the whole universe of potential other things.
Yeah, well, thanks for that question. So this is a super important question, right? So we're discovering all these chemical resources. What are we doing with them? They're sitting in our freezers, right? And we may run some simple E. coli screen. Thank you, Joe. That's your strain. We were actually using that assay. Because, we can do it and it's easy, right? And then we have collaborators. The cardiomyocytes are collaborators at UCSF who it's amazing. They use stem cells to transform them into heart cells, and then they feed. It's crazy. So it's just random collaborations, right? It's not done in an effective way. So we're starting to try to build some compound libraries that we want to make available. Bill Gerwick down at Scripps, one of our colleagues, has started to do this and built a compound library that's available to everyone on campus if they want to screen it in their assays. NCI's invested heavily in trying to build libraries and make them available, but it's a problem, and it's been a big frustration for me personally because, hey, we could have an AIDS drug sitting in our freezer that's better than anything on the market, and we don't know it because molecules aren't being screened effectively. So if you have any ideas how to do this with Novo Nordisk, I don't know, but it is a real problem that molecules don't get screened effectively.
And Jack, one other thing, before we are super interested in brain inflammation. We want to see how to pursue that. I wonder, yes, you show some really fantastic data in neuropsychiatric. Is there a way to look at neurodegenerative and inflammation in the context of metabolic, such as obesity?
Oh, yeah, absolutely, there is. We can definitely do neuroinflammation assays in animals. Obviously, I've tried to do them in humans, and nobody wants to give up their brain for testing. I'm not entirely sure why. But either way, it's a definite issue. But we can drive down neuroinflammation, and that relationship is incredibly important, right? I think of depression, neurodegeneration, certainly neurodevelopment in utero as an inflammation-associated disorder. And so understanding that narrative, I think, is key. One of the key points is, what is the mechanism of action that's driving that? We have a new model system for lung, for example, where we can use lactobacillus producing indole-3 acetic acid, which is an IL-22 mediator via eosinophil activation and macrophage activation, which is incredibly potent at driving down inflammation and improving barrier function in the lung. Treatment for COPD, for emphysema, right there, right? But I just wanted to tap on what you said and what Paul said. Again, finding these assays and getting these assays in line is incredibly difficult to do. and making sure that they're all available to the researchers that have them, and the funding's available to facilitate that screening is something which needs to be worked on at the federal level. I would fire the question back at you. Paul and I, working with Brad Moore and the rest of the CMBB team at SIO, are building a biological automation facility, which will have a screening potential in there. But what are the IP barriers that would prevent a big company from coming in and saying, okay, let's access your library and screen it for all of these components, right? Is it, I know universities are very IP protective and therefore my lawyers won't let me get involved? Or are there ways of mitigating that concern and fear that would facilitate actually making these resources more available to private industry? It's a question.
Yeah.
Ask the question.
That's a very good question. We definitely are looking at different, I mean, it's hard, it has historically been difficult, but there are ways, and we are, in a new sort of era of looking at different models, and universities and the private sector and pharma are really open to think different. So I think there's definitely a venue to.
Well, an IP-free zone then, like, we carve out an innovation unit like Georgia Tech does, and say that anything that's created here we will waive IP rights on. Would that be beneficial? I mean, to a certain extent, we need to ensure that IP revenue comes back to the university. That's incredibly valuable. Nobody wants to pass up on the next $1 trillion drug, right? However, as Paul says, when you've got this much potential sitting in a system and you can't do anything with it without that partnership, that's what needs to be broken down.
Yeah. We, have been doing things in that space. We just signed with the Broad Institute a really interesting collaboration that deals with these things so it's something off a microphone that we will continue talking about. Definitely there's things that we need to, if we want to see this progress, and we just need to figure out the way of working together.
Okay, well, you guys know each other.
There was a question back there.
You can probably shout.
While you're thinking of it, I actually wanted you to build off what the New Center we're building on, was your vision there. To what extent are you building in deep learning and generative AI in that solution?
One of the things that we've been dealing with generative AI especially is facilitating more effective means of allowing communication with the data set, right? So building a data set and then creating almost a generative AI simple GUI to go on the front end of that data set to allow us to ask effective questions. A lot of businesses are developing this strategy to allow their HR departments and their innovation departments to actually ask questions of data within an organization. We have enormous quantities of data in our system, climate data, environmental parameter data, biological data, genetics and information. Sometimes we know the question to ask, but we don't know actually how to do it. So if we can use generative AI to actually ask the simple question, please find me all of the genes here that are also associated with gene Y, right? Then we can use deep learning as the back end to actually facilitate that interaction. So that's something we are trying to embed in our process at SIO, trying to embed in the process at UCSD through our HDSI Data Sciences Institute. I don't remember the name. But that effort is at the forefront of what we're doing, but it's still very much in the research space, right? It's still clunky. Anyone that uses ChatGPT 4 will know it's clunky, even if it's quite remarkable. So there are ways and means to do that. And the impact on biomedical sciences will be phenomenal, right? The ability to access a patient's data and compare it to tens of thousands of like patients within a population database will be phenomenal. You just need the doctors to be able to ask simple questions and use deep learning on the other end to actually do the analysis.
Yeah, I have a bunch of questions. There were 2 talks and they kind of led to different questions, but just to follow up on the AI point you just made is there have been papers, I think in Nature, there was a material science paper where they had a model crawl all of the material science papers ever published and learn the structures of different materials with the desired properties. And then they were able to predict the structure of materials with the desired properties that they wanted using generative AI. And it seems like chemistry and your library would enhance an AI like that incredibly. Paul said something about being able to predict the compounds made by the genomes. I actually didn't know that was possible. That's pretty wild. But now you've taken the reverse approach where you're looking for compounds. Can you then go back to your metagenomics and figure out which organisms made that compound? And then make predictions about what the function of that compound is a more rational way to filter through these libraries. And then I have a second question, completely unrelated, about your at-home microbiome testing. As a hearing loss researcher, I fantasize about getting more measurement from the population about what kind of noise they're exposed to and what kind of hearing loss they have over time to identify genetic causes of hearing loss. But it seems like it would be really expensive to deploy devices to homes and have all these measurements made and analyzed. And I'm wondering, how do you go about getting that kind of project funded and supported?
Well, we'll answer the second question first because Paul just passed me the microphone. But a lot of what we've been trying to do is reduce the barrier to accessibility to technology. So deploying these technologies into people's homes in a bulletproof, simple to use way that's hard to corrupt, hard to deal with, hard to destroy, right, is really important. One of the things for stool collection that we found is that reducing the cost of generating the data was the key concern of our customer base. So for BioSense, for example, we've reduced the cost of doing metagenomic analysis by 90%. And it's better or as good as the best microbiome analysis that you could possibly do, which is having somebody come into your molecular biology laboratory, poop in a cup, and extract the DNA right there and then. It does everything right there and then. It removes all of your molecular biology costs, all of your shipping costs, all of your soil sample costs, all of the errors and biases associated with humans. That automation, I think, is key. How do you get that funded? You get that funded by demonstrating that there's a financial model which will actually support it, right? And so gathering data is key, but there has to be a financial incentive to enable it to happen. Apple does a lot on, and my voice tends to activate this noise detection system a lot, as Paul will attest to. But that I think is an incredibly key thing. You have to build these practices into what people use every day, and then enable them to use it without even realizing that they're doing it. And hence the reason putting something in a bathroom where people wipe their bum and put a piece of used toilet tissue directly in the device, that's simple. It doesn't require any additional aspect that they have to deal with, and people can adopt it as an everyday technology. And the first question?
Right. So we've learned a tremendous amount about the genetic basis for natural product biosynthesis. And, we think DNA, RNA, protein does a job, game over, right? But there's another step here because in this case, we're looking at collections of proteins that sequentially build these small molecules. And these collections of proteins are encoded by some of the largest genes known. And we're talking about clusters of genes that can be hundreds of kD. And so these are huge gene clusters, and there are now bioinformatic tools that can recognize them and make predictions, not only about what class of molecule will be made, but also in some cases, what the structures will look like. So there's a lot of opportunity to use AI and big data sets to build on this, but it's really an exciting time, and it's allowed a lot of people to get into the game of thinking about natural products because they can run their data sets through these pipelines and make predictions. The harder part is you raise functional predictions. And so functions in nature are very hard to understand. We know in a few cases certain molecules are involved with iron chelation, and that's been shown experimentally. That's pretty straightforward. It's really difficult when we're thinking about biomedical applications to relate that to what the functions in nature may be. But there is some very clear evidence that molecules that have potent antibiotic activity in our assays are actually functioning that way in nature because we often see resistance genes in the gene clusters. And that's a super cool way to predict the target of an antibiotic before you even find the antibiotic, because you see the resistance gene, and there's a gene in the biosynthetic gene cluster that's making a small molecule, and then there's a housekeeping gene in there. You're like, why is that housekeeping gene in there? And then you go, it's a second copy, there's another one, oh, there's a mutation in the active site in that housekeeping, in the protein encoded, and bingo, you've got the target. And so looking for new targets is super important, right? And, you could find an orphan gene that's highly conserved. No one knows what it does. Bingo. Now you could have an antibiotic acting on a new target. Otherwise, though, really tough to get at the functional role of these molecules in nature.
Another question, but I just wanted to have you guys step back and talk about your experience with funding, finding the right kind of funding, what you think is the future of funding. And you have your responsibilities cover a lot. You have administrative, research capabilities, and you're both active in implementation, so in the development process, and you have startups. So how are you doing, who is doing the funding? What are your thoughts about how you can have such broad reach? And where are you getting the sources, and who's kind of helping get that heavy lift?
Academia really does self-select for self-actuating people. You're right. Especially if you're at an organization such as Scripps Institute or JCPI, for example, where you have to cover all of your salary, it's incredibly motivating, right, to go out there and submit a hell of a lot of grants. And I think that's one of the key things which drives us forward. I have 22 people in my laboratory research team that I have to make sure that they're fed and watered on a regular basis, right? Makes them sound like horses. But you got to make sure that that people have income, and so that is really a strong motivating factor. Where you do it, the federal government's become extremely good at facilitating large-scale programs that allow for big product development, and the ARPA-H program, the new federal program, is incredibly valuable. There's a lot of money in that program. It's highly innovative. It allows you to do things which you wouldn't be able to get funded through a normal strategy, because they're not looking for somebody who's got a solution incredibly baked. They want something that's cutting edge, but they also want incredibly tight milestones. Right, it's like a startup company and an investor. It's a relationship that most academics aren't used to, but if you can actuate those opportunities, it's incredibly valuable. And I think that's where a lot more of the funding needs to go, towards demonstrating that you can support large-scale operations, you can build programs that are flexible and have opportunity, and ensure that they can deliver upon the framework of research that you think is going to be most important. But I don't know. We have donors, we have federal government, we have the state government, we have SIO, we have tens of millions of dollars in line items associated with the California state government, because it's important to maintain a lot of the processes that we're doing. They have real value politically and socioculturally for the people in this state. And so that's a key aspect, trying to bake these research programs, these innovation cycles into the state and federal legislature is incredibly valuable for us.
From the infectious diseases standpoint and rising antimicrobial resistance, it's really exciting that you're looking at oceans for new molecules. What is discouraging, and I don't have a solution, I just want to highlight the problem you've already highlighted, and has been highlighted in the morning sessions as well, is that even with molecules that have made it to have been FDA approved or are at phase 3 level, companies have dropped them because they need to make enough money from that. So I think it's worth keep highlighting this issue till we get over it, because there's no other solution for antimicrobial resistance. Two other things that I wanted to, kind of just ideas that I wanted to bring to the group's notice. One is climate change. So with climate change, we are going to start seeing diseases that were not there in certain areas start appearing. For example, we're already seeing valley fever, coccidioidomycosis, that's spreading to areas that are becoming drier now. It's there in the soil, it gets stirred up, and it's expanding. Things like malaria, you have mosquitoes in newer places, and it will spread there. So sort of, how can we use technology to predict those, to detect those, and then, of course, intervene in those areas. The second idea was about microbes. I make a living by finding effective ways of killing microbes. But I was thinking we have been using microbes as treatment and using them for behavioral issues. I think that's an excellent idea. But we've also been using now replacing the gut microbiome in patients who've been on antibiotics, to treat C. diff infections. We've been using microbes such as Mycobacterium bovis to treat bladder cancer, and we've been using them as vaccines for a long time. But I think one area may be looking at people who have been on antibiotics and their gut microbiome has been wiped out. And then once it repopulates or you repopulate it with specific bacteria that you're interested in, and see whether that affects their, say, depression or other issues. There are other microbes that do have an effect on the brain and behavior. For example, toxoplasmosis. That's one of the conditions that I've been interested in, but that seems to have more of an impact on behavior itself besides psychiatric illness.
If I'm a mouse or a rat, I'm going to start being attracted to cat urine. Maybe we can adapt that to humans. Yes, there are lots and lots and lots of ways of modifying the microbial community in order to facilitate health, right? We have a lung microbe axis, we have gut-brain axis, we have cardiometabolic disease, we have immune diseases. We work with engineering here. We have a large NSF program at SIO that's focused on building biologically active building materials that have immunogenic activity, right? So you can put them into a school and have children physically interacting with surfaces which will stimulate immune responses and reduce long-term atopic risk in a population. Works in mice, got no idea if it'll work in humans, but it sounds cool, right? We'll build living buildings. So those kind of things I think could be incredibly valuable as well, shaping the outside as well as the inside of people. And I can't remember your first question now. Do you remember the first question?
This is the effects of climate change.
Yeah.
And I would think we want to talk about aerosols as well, and what we're doing to help kind of connect what you can anticipate is happening because of climate change and the impacts in public health.
Yeah, and that's a really important point. But the environmental etiology of disease angle is incredibly valuable, right? And people have been doing more rigorous testing at the state level. The Wadsworth Center in New York is a really good example of this, of animal disease spread and the range distribution activation associated with climate in order to understand, get in front of the epidemiological consequences of that range diversification spread. But it's something that stopping is going to require better vaccines, better strategies for environmental control, like we do with malaria, and adding in organisms which will make the mosquito sterile, right? So there are opportunities to explore that could facilitate with that interaction. So aerosols are a key piece. We have a fantastic researcher, Tariq Bedmania, down at SIO, who's focused explicitly on dust, and especially in the Imperial Valley at the moment, which is becoming an incredibly aggressive problem for respiratory diseases. As you pointed out, some of these organisms that are present in the soil are being reactivated, aerosolized, breathed in, and they're hitting our most vulnerable communities first. And that's a major concern and something that needs to be dealt with. We also have researchers like Kim Prather who are focused on trying to understand if things in the ocean are getting into the air and affecting human health. We don't have any data to show that at the moment, but it's important research that needs to be done.
Let's stay with climate change for a minute. 90% of the excess heat over the last many decades has been absorbed into the ocean. And so what are your concerns about the impacts on the biodiversity, microbiodiversity, and kind of really not solutions, but what are your thoughts about how urgent is it to be able to understand our oceans now, given that heat absorption?
Yeah, it's hugely urgent. We're talking a lot about the human microbiome, but we apply the term microbiome now beyond humans, the oceans microbiome, soil microbiomes, and, there are soils that naturally suppress diseases. We call them suppressive soils. That suppression is microbially mediated. And then there are soils that are susceptible to disease because they don't have the right microbes. And the reason they don't have the right microbes is because how we've treated those soils, right, with fertilizers and other things. So these are big things to think about. I mean, the things I get concerned about are, climate change is anthropogenically induced. We're doing a lot of things that aren't just affecting CO2. We're talking about mining the oceans off the coast of California. We've been working on a project out here with my colleagues, Lisa Levin, who's a deep sea invertebrate zoologist, documenting the animal and microbial communities that live in the deep sea in these mineral-rich habitats that might get leased for mining. Because if we don't get baseline data on what's there, we're not going to know what was lost after these mining activities occur, right? And we're looking at what's there. We're also looking at their biopharmaceutical potential, right? Because there are lots of opportunities for taking these concepts to the deep sea and making discoveries. And once all this mining starts happening, game over, right? And everything changes. And so, this is another type of climate change. I guess not climate, but environmental impact that's really affecting our planet. And so getting baseline information is hugely important. At least that's something that, I think a lot about.
Let's very briefly augment that. I mean, and to Paul's point, we need to look at the microbiomes of corals. If we want to maintain corals, we have to actually understand the microbial partnerships and the relationships which facilitate them becoming more resilient to temperature, acidity, and other pollutant threats. We have a large program here working with Jen Smith focused on regenerating our seagrass habitats and our kelp habitats in order to facilitate those. It actually has a microbial component as well. We've been exploring how to restore mangroves, which are incredibly good at both capturing carbon dioxide, but they're also fisheries for nurseries for fisheries around the world that can support local communities. And using that, we've been looking at ways of adding probiotics into the soil, fungal and bacterial, that can promote the restoration and resiliency of mangrove trees to pollution. So there are so many ways that we can actuate the microbiome in the environment in order to both control pollution, control disease, ecology, and ensure that those environments remain productive. I think the other piece is the one thing I'm most concerned about with climate change is the environmental etiology of disease coming out of the ocean. So Vibrio parahemolyticus rates are going up across Southern California and in Florida up to Georgia, and they are nearly directly correlated with sea level temperature rises. And so as we start to rely more on aquaculture for our protein and our nutrition, we're going to have to understand that relationship. And this is something which Rita Colwell, who was one of the NSF directors that founded the Environmental Etiology of Disease program, pointed out 30 years ago in Bangladesh that Vibrio was increasing in the waters and it was making people sick. And we are still only just starting to understand the impact of that on the United States. But it is a real threat to our economy because people get sick and then they take many days off work, as well as to people's overall health.
Before we ask you all if you had any final questions and thinking about that, I also wanted to mention that we do have some integrated programs, and you two might also want to add to this. But I'll mention Start Blue, which is a early stage accelerator that is based here in San Diego, and it's managed by Scripps and Brady School. And we would love to have people coming from health sciences or participating. It is a cohort-based. You have to apply. We're going through our third cohort, which you can track it, look at what's coming, and participate in some of the open discussions, and then learn how to apply. We have people that have gotten I-Core support that have come through Start Blue as well, so it applies to anything that touches or related to oceans and water. And we have an extension that I'm going to leave to you, Jack, to mention that's coming from NOAA related to climate resilience. And we're in the first phase right now, and this would be also another thing across campus that we can have some collaborations.
Yeah, so this extension is led by Vanessa Scott, and I'm the nominal head. But the plan is to deliver upon climate resiliency solutions for ocean technology that can facilitate development. So this will actually come in the phase 2 with startup funds. So the opportunity to seed fund small operative organizations with enough capital to get them up and running and then connect them to the relevant folks who can help to lead that program forward. And I believe you have similar programs that are starting up in biological sciences, Kim?
Not quite so far along.
Yeah, but it's but UCSD generally we're trying to explore that ecotype. Operating these kinds of budgetary initiatives to create seed funding is incredibly valuable for the university. And most of our students now are going into industry, right? You know, the money is real, and they're seeing the writing on the wall. Let's say about 70% of them end up in that direction. And we want to support them in doing that. I've got 2 of my graduate students who are looking to start their own companies in the next year. And so that's an incredibly valuable route, right? Creating the ecosystem that will facilitate them being the next leaders at that scale.
Very good. Thank you. Very good insights. I just had a couple of points. One is the biome sense. You know, if it's a very interesting idea. If students want to prototype their ideas here on campus, is there access to biome sense at UCSD campus?
What, the technology itself?
Yeah.
There is, but I would have to understand what. So we can put them into hospitals, right? That's probably the easiest strategy. And they're currently being deployed in 3 clinical trials here at campus. 1 clinical trial in Chicago, 1 clinical trial in San Francisco, in order to generate data. But really at the moment, the key investors are interested in it from a human health perspective, and that's where it's being mandated. But if students were interested in leveraging that kind of technology for their own research programs or own innovation opportunities, that's something we could talk about. Okay, so you're envisioning it as an at-home device, right? Like a B2C, or is it more like hospitals? I mean, the money, to be perfectly honest, is in a recharge through insurance, but predominantly in clinical trial development. And so, a big pharma organization that really wants to come in and add microbiome data into their clinical trial to prevent their phase 3 trial or phase 2 trial from failing because they haven't accounted for the variability in the human population, which led to a very, very low N and therefore very low statistical significance for the outcome. We think there's a real potential there to better characterize populations and say, look, this drug will work in that population. I really do think we need to move away from we have a drug that works 80% of the time, but only in 20% of the population, and more towards drugs that work, what's it the other way? Yeah, we need 20% of the time in 80% of the population, we need drugs that work 80% of the time in 20% of the population and diversify in that way. And it needs a better economic model to facilitate it, but we do need better stratification of our human populations before we start running clinical trials. Because I think we're leaving a lot of drugs on the table when these clinical trials fail. I mean, that's a major concern that I have for the industry moving forward. So my second question is about the business model. This morning there was a discussion giving the kickoff about antibiotics and the challenges, and Dr. Ajay alluded to that just now. Similarly, on the IP side, there was a lot of discussion in the morning, but when you're dealing with ocean and environmental resources, have you found any clever ways to both deal with IP, because investors want IP, right, in order to fund this stuff?
Yeah, sorry, I missed the discussion this morning, but in the natural products world, you used to be able to obtain a composition of matter patent on a new structure that had an activity, and those rules changed, so you can't do that any longer. And so it's become more challenging to do natural product drug discovery in that sense. And so now you have to just try to find a use for a molecule and patent that use. And so it's a little more challenging in that sense, and it's taken some of the clear path. It used to be a pretty straightforward thing. If we found a new molecule with an activity, we'd immediately contact the Office of Innovation and Commercialization and file a provisional patent. And now it's like, well, we can't patent the structure any longer, so, there's more barriers. So there are some new challenges in that area for sure.
I'll just add one thing to that. Innovation in the oceans is a little bit easier because a lot of our samples are collected in international waters. And while there's federal protections, if the money used to collect it was federal, there are ways and means of facilitating access to those samples that do not require can overcome legal barriers associated with material that's collected in sovereign land.
No, I was just going to make a comment about the developing IP from a naturally derived small molecule. So typically, those molecules will oftentimes have properties that aren't really good for pharmacologically. And so you need to derivatize those, correct the solubility, plasma protein binding, all these types of things. And so I think there's still a large scope to do that. Of course, you need some chemists and ag chemists to look at your molecules, find the things that pharmacologically maybe are red flags or things that would improve them. So I think there's still a tremendous scope to do it. But you're right, you do need to diversify the team or find other partners to help you do that.
So I think we're gonna be wrapping up.
One last question.
Okay, one last question right here.
Hi, thanks for the talk. You guys mentioned this in the microbiome company earlier, which is, I'm assuming one of the bottlenecks for the science is sampling, right? If you go sample. Aside from direct to consumer, like what are the ways that you guys think that is being solved?
It's not at the moment in an effective way. I mean, so the best that people have come up with are creating wipes that people can use at home and then ship the wipes in. The problem is you still have shipping costs. You then have variation in how the laboratory processes the samples. The bottleneck is getting people to give fecal samples in a way that doesn't make them feel icky. And it's really variable between different populations, right? Different cultures, different groups like interacting with their stool more or less. I know that sounds weird to say that, but there are poopophiles and poopophobes. But that really does change our ability to access certain populations. So, for example, in our Native American populations, there is a recalcitrance towards collecting stool or physically interacting with stool. And so this removes that recalcitrance. And those barriers, the technologies need to account for those social barriers as well as the technological barriers. That's really important.
I just want to encourage everybody to reach out to these two or to the Center for Marine Technology, Biotechnology and Biomedicine, or to a woman who we keep referencing, she's not able to make it today, is Vanessa Scott, because she runs our Corporate Alliance of Innovation. She can help you navigate strips. So come on and navigate with us. And again, pleasure to be here. And thank you guys so much for taking Saturday and Tuesday.
Can I catch you real quick? Okay. I have no expertise in this. You're on the P side, I'm on the geriatrics.
Oh, now we have a short break, coffee break, and then we are off to the next panel, which is partnering for resources. We have two speakers, one from Novo Nordisk, who's already had questions, and another from a venture capital company, Sandbox, who's coming down from San Francisco. So please be back at 2:15.